Basically, I've seen that this section is mostly on sarcopenia, and I will rather talk about cachexia, which is another form of muscle wasting. Very likely this is actually fasting, not really cachexia, this young lady here. Anyhow, the two types of muscle atrophy have something in common, but of course, major features that are different. For instance, I am a histologist, so of course I look just at the face of the muscle, and things such as the fact that atrophy is heterogeneous in sarcopenia, and also the phenomena like grouping and the switching to slow phenotype. This is definitely something that is distinguishing sarcopenia, and is not really happening in cachexia, that is characterized by an abrupt and generalized muscle fiber atrophy. Nonetheless, the two conditions have something in common, including one of the most important features, that is frailty and muscle wasting and dysfunction, which is so important for the quality of life of these people. And of course, also given the indirect effects of muscle contraction on myokines production, for instance, for the response to therapy, the well-being, etc., etc. This atrophy is due to is associated to metabolic impairment, characterized by an increased catabolism, especially with protein. All this leads to morbidity and definitely to mortality. Remember, of course, everybody by aging is dying, is gonna die, 100% of the cases, but remember that about 20% of the cases of cancer patients, they die of cachexia and not of the tumor. There are, of course, major avenues to fight cancer cachexia. The fact that there are still many possibilities here basically demonstrates you that there is still no one single thing that it's working. So there is not a cure for cachexia to date. However, physical exercise is a very promising treatment. And since this is basically having a mechanical effect of muscles, we are pretty much interested in the pathways that are activated by physical exercise. In particular, we have been focusing in the last few years on the role of serum response factor, a transcription factor, as you can see, thanks to the nuclear localization signal here, that it's involved in many things. It's in general, it's very good for the muscle. Like it controls satellite cell function, expression of major muscle, skeletal muscle genes, and so on and so forth. And guess what? It's decreased in conditions when you don't use your muscles, such as immobilization, aging, and as you would see in cachexia. Let me also point out that it works in the nucleus together with a cofactor that is called MRTF-A, which binds to SRF and helps its transcriptional activity. Now, well, just still the last slide of background, SRF and its translocation to the nucleus along with MRTF is actually activated by mechanical signals. The signal is going through the STARS activity and RAW-A, and they are kind of linked to the polymerization of actin that is induced by muscle contraction and mechanical stimulation and so on. And this ultimately, the fact that G-actin disappears allows the cofactor to get into the nuclei and basically pushes SRF transcriptional activity. This has a result on muscle fiber metabolism, as I told you, but also through the secretion from muscle fibers of interleukins, such as interleukin 6 and 4, it promotes satellite cell proliferation first, and then fusion and recruitment to the muscle fibers. So remember, we are always in love with this idea that we don't have to look to the muscle fiber metabolism alone, but also to the surroundings of the muscle fiber, to the niche, to what's happening around the muscle fiber. Now, let's have a look to SRF. I will show you first some data on human patients, cancer patients that we had through our collaborators that I have to acknowledge and thank immediately. So these are basically three groups: the group of Professor Muscaritoli at Sapienza in Rome, the group of Sorci and Riuzzi in Perugia, and then the group of Marilia Silender in Sao Paulo. The advantage of the last group is that they are also having patients that are that do exercise. So we will see what happens then. However, let's start having a look to SRF in these cachectic patients. And as you can see, there is actually an accumulation. Here you see the quantification in red of a band that represents basically the degradation of SRF. I will not have time to develop on this, but I will be glad to get questions on that. So basically, SRF is cut, is degraded, is downregulated in cachexia, and indeed, its transcriptional activity is decreased. SRF gene, it's a target of itself, but also other genes such as Rho, integrins, actin, etc., are all downregulated in cachexia, meaning that SRF transcriptional activity is decreased. And we believe that this is important because the amount of SRF degradation, the degraded form of SRF, actually correlates with the extent of the weight loss in these patients. So it might have a clinical relevance. The hope is not lost, though, because as you can see here, in the presence of exercise trained mean that this patient have done a protocol of exercise, basically endurance exercise throughout several weeks. And the running activity basically induces recovery. Let me actually move, let me move this thing. Induces a recovery of the SRF expression in terms of RNA expression. So RNA expression is increased. And the interesting thing is that basically here we could compare cachetic patients and patients that have the tumor, but they are weight stable. But I will not further develop on this. For now, the message is basically that the exercise might have an effect on SRF expression. So then to better manipulate the levels of SRF, we went to the mouse model, tumor-bearing mice that are subjected or not to free exercise, free physical activity consisting in wheel running. And these are data that actually were kind of known, the positive effects of exercise, of wheel running. You see the body weight loss in the presence of the tumor, and wheel running rescues this weight that correlates with the rescue in the muscle weight. And of course, this is it goes along with the cross-sectional area that is decreased by the presence of the tumor, and no more significantly lower when the mice do exercise. And of course, this goes along with the molecular markers of muscle wasting. So the ubiquitin ligases that are ultimately responsible for a major pathway of protein degradation, they are both upregulated in the presence of the tumor, and they go back to normal when the mice do exercise. Now, of course, we are interested in the mediator of the beneficial effects of exercise, and we wanted to test whether SRF may play a role in this. So we looked at the levels of SRF. This is a Western blot, and basically you can see that similarly to what happens with humans, the full band of SRF goes down in the presence of a tumor. This is in mice again. And it stays high in the presence of wheel running. Again, this is another way to show you the correlation between SRF and the loss of body weight, meaning cachexia. The lowest SRF is, the highest is the loss of body weight. So there is this very linear, very clear correlation between the levels of SRF and body wasting in terms of very general way of summarizing and looking at a bed state, at cachexia. So SRF levels are important. And as you can actually, you might start noticing it here also, but it's also visible here in multiple Western blots that we did from multiple experiments. You see that basically, while we consistently see a decrease of SRF in the presence of the tumor here and in this experiment here, etc., when mice bearing the tumor, they do wheel running, the levels of SRF are variable. You see, like in this case, in this experiment, the duplicate mice here, one responded with very high levels of SRF in the presence of the tumor, and the other one was looking as if he did not anything in terms of exercise. This was a very bad experiment, very frustrating for the student that did it, you know, because basically the mice exercised for weeks and the tumor and everything. At the end of the experiment, it didn't work. Okay, what does it mean? It didn't work in the sense that we didn't see a rescue of SRF expression. And the worst thing is that actually these mice were also displaying, as I told you before, a loss of body weight. So they were cachectic. So, I mean, we could not cure them with exercise. And here is the more complex situation. You see the mouse that had no SRF was here, losing weight and being cachectic, and the mice having SRF was here. So, of course, we did this multiple times, multiple experiments. We were kind of desperate, and we exploited this variability, this fact that this exercise was done at will. So some mice actually did a lot of exercise, while other mice did not run enough. And here is the summary. Like, this is just four examples that you have seen before. Of course, we followed every single mouse almost every day to monitor that how much, if and how much he was running. She was running, because they were female mice. And as you can see, basically, the only mouse displaying high levels of SRF, even in the presence of a tumor, was the mouse that was always doing a certain amount of kilometers per day. So we basically did this for a bunch of mice, and we could correlate the amount of exercise per day with the amount of SRF expression in the presence of a tumor. And thanks to statistical analysis, sorry for the little error here, the H that is missing, we basically found the threshold of the minimum amount of exercise that was needed to keep SRF high, and this correlates with the rescue of for instance, body weight. And this amount of exercise for a mouse is minimally 1.5 kilometers per day. Very often, actually, let me tell you, the mice run much more than this, including the cachectic and tumor-bearing mice. They love running in the cage. They do, let's say, around 4 or 5 kilometers per day, the sick mice. The other one, they do twice than that, which is, of course, much more than what I do, and except Feliciano maybe, or someone athlete here do. Let's go back to the average situation. So in general, the mice run, and so we average the amount of SRF and the response in these mice. And this is, again, the average of the whole population. We looked at the SRF-dependent genes, in the sense that here we're looking at the expression of genes that are targeted by the SRF transcriptional activity. We looked at a bunch of them, and basically the result, I will not go into the details, it's about the same for all of them. So for instance, let's say stars, which is important for SRF activation. This is the control. Half of its expression when the mice are bearing a tumor, and this is going back even higher to control when they do, in spite of the presence of the tumor, while running. You know, so this pathway is activated. Integrin, it's a target of SRF, goes back, and the interleukin that I mentioned, you see 0 point something in for both when the mice have the tumor, and they go back to the control level, or even twice the level of the control when tumor-bearing mice do with running for interleukin 4 and interleukin 6 respectively. So I will cut a little bit of this, but basically SRF transcriptional activity is rescued. Now, the question was, is SRF necessary for this response to exercise, to the muscle contraction and the rescue? So, of course, to prove the necessity, we did loss of function experiment through the creation of a tamoxifen-inducible and SRF knockout mice in the myonuclei, and so in the muscle fibers. Here, we had to change the tumor to be compatible with this strain. So we used the other classical model of tumor inducing cachexia, the lung carcinoma, and this is just a preliminary experiment showing you that our knockout mice are really SRF knockout mice. Another preliminary experiment that we did, and we published in European Journal of Translational Myology, which is therefore validating our discovery as the greatest of the world. The other experiment that we did was to double check that the SRF knockout mice were able to behave like other mice, like wild-type mice, when they do exercise in terms of, for instance, speed and VO2 consumption, etc. Because, you know, SRF is so important for the muscle function that we wanted to make sure that these mice still can exercise and adapt to endurance training like the others, you know, in order to avoid to have a biased result. So they behave like the wild type. So we did the experiment. And now in the next experiment, please be aware that we still have 4 populations, but these are all mice bearing a tumor. It's not with or without tumor, with or without exercise. Here, all the mice are bearing the tumor, and then the 4 population are either wild-type mice that are doing exercise or knockout mice that are doing exercise. So the fact that all these mice are bearing the tumor means that this tibialis is cachectic, okay? So this
4 minutes left.
Okay, this mouse is bearing a tumor, and this is the mouse that is cachectic. Exercise increases the mass of the muscle, and this does not occur anymore in the knockout mice that are bearing the tumor. So the rescue by exercise is not working anymore. And this is the cross-sectional area, no rescue. And this is the loss of the increase of atrogene 1 when the tumor is there in the wild type, no, and the decrease when they are doing with running and it's not working anymore, and so on and so forth. Therefore, SRF is necessary to mediate exercise effects in cachexia. I wonder, maybe I will just show you this last slide based on the fact that we have previously demonstrated that PAC7 and the surrounding satellite cells are also involved in muscle homeostasis. I will show you the effect of exercise while running on some parameters of the fibers in terms of cells. This is the number of nuclei per fiber, per muscle fiber. There is a decrease in tumor-bearing mice. This goes back in the presence of exercise. This is a marker of cells that were proliferating, the EDU, that it's incorporating in proliferating cells. And then we looked at the muscles and at the nuclei that are incorporated within the muscle fibers. We sort of normalized by the number of EDU positive nuclei in the section. And as you can see, we counted what's out, what's in. Most of the cells that are proliferating remain outside of the muscle fibers in the presence of the tumor, while the situation is totally different when exercise is done. And the PAC7 cells that are increased in the, remember that PAC7 is a marker of quiescent cell, but it is inhibitory for the differentiation of the myoblasts. It stays high in the presence of the tumor, and it goes back to normal in the presence of wheel running. I will skip the in vitro part, and I will just maybe summarize some of these slides, basically showing you that cachexia decreases the expression of SRF and its transcriptional activity, while exercise, a minimal amount of exercise around 1.52 kilometers per day, it's enough to rescue this activity, which ultimately leads to muscle homeostasis. And a similar regulation seems to occur also in human. Again, I will not develop on some hints that we had on the mechanisms of SRF downregulation due to lack of time. So thank you very much for your attention. Thank you very much to all the collaborators. This is a little mistake. I wanted to write Alexandra Backham here, but all the students that participated to this work, as you can see, they are named Alexandra in either language here. So we have many Alexandra to thank, and Mehdi and Katerina, who did most of the work. Thank you very much.
For this very interesting presentation. The paper is open for discussion. Are there any questions from the audience? Okay, if not, I have one. You shown that there are two degraded form of the SRF that are increased in cachectic patients. Are their behavior differently? I mean, the two different isoforms, the 55 kD and the 70 kD, have different expression levels that to correlate maybe with muscle wasting or with the interleukin-6 secretion?
Yeah. Basically, thank you for the question, Alexandra. I was sorry, Sandra. I'm still obsessed with the fact that the wrong Alexandra was written in the authors. Yes, basically what we notice is that in cachexia, and correlating with the weight loss in humans and in mice, there is an increase of this band that is smaller than the regular one, that it's not transcriptionally active, and that represents, and you see it here, the quantification of this increase. And by the way, RNA level does not really goes down. So SRF keeps being transcribed. That's why probably this amount of SRF is still there, but it's immediately degraded. As a result, its transcriptional activity is, and that this 55 isoform is not transcriptionally active has been demonstrated by others, but by some molecular biologists. And what we notice is that the accumulation of this form here, it's associated to a decrease of the transcriptional activity. And then in vitro and in vivo, we have partially demonstrated that caspases are actually responsible for these hydrolysis.
Hydrolysis, okay. And are there any data in the literature about the SRF expression in human cachetic patients?
There is some, Judge actually has done some work on that. He did some omics analysis, and I'm pretty sure that they have observed that it goes down. SRF expression goes down, I'm pretty sure. Unfortunately, I guess he's not here, right? Because of the time zone.